record. Okay, sorry people, technologically challenged I am. So now we should have a presentation. All good? Yep, great. Okay, welcome everyone to Coast to Corals online edition. Our wonderful presenter Ilya is not here tonight. She's somewhere in Africa, potentially on the road somewhere. So we wish her good luck in her travels. Firstly, we'd like to acknowledge the traditional owners of the country on which these presentations are coming to you from and where you are. So because we're spread out all over the country, that will be a different traditional owner group depending on where you are. Because I'm up in the Whitsundays, we have about 5 different groups that identify with this area. And I'm sorry, I can't remember them all offhand, apart from the Nauru and the Juru. But we acknowledge all of their elders, past, present, and emerging. Tonight's online event, well, you should know what it is because you're all here. It's the wonderful Dr. Asia talking about the biodiversity of Lady Elliot Island. So who is Reef Check Australia? Well, we are a not-for-profit citizen science organisation, and we're dedicated to helping our reefs by empowering ordinary citizens to get involved. So we conduct reef health surveys pretty much all the way from the New South Wales-Queensland border all the way up to Low Isles, just past Port Douglas, and everywhere in between, depending on funding. We also have an amazing group of people called ambassadors who educate the general public on what we're doing, the health of our reef, and how they can help even by simple acts of changing their habits at home. So a little bit of housekeeping, online etiquette. Most of you already have your audio on mute, which is wonderful. Please feel free to use the chat. Got Jodie on there, she can talk to you as we're going and I can help answer any of your questions. We'll do questions at the end, but if you've got any pressing things you don't want to forget about, pop them in the chat and we can run them to Asia at the end. If you're not on the mailing list and you found out about this on socials, you can email me on seqsurveys@reefcheckaustralia.org and I can put you on our e-newsletter list so you know what we've been up to and what we've got coming up. Plus we've put in some little book reviews and little podcast reviews of things we've seen that we think would be great to share. Upcoming events. So I went to this, the Ocean Film Festival in Proserpine the other night, and it was really awesome. They're showing in Noosa on the 15th and 16th of March. We will have a team at the one in Noosa on the 16th of March, so please pop in and say hello to them, ask them lots of questions. Test their knowledge for us, that'd be great. It's on in Townsville, 16th, Cairns the 17th, and Gold Coast the 29th and the 30th. Unfortunately, we couldn't get into Townsville or the Gold Coast because they were booked out with other conservation groups. Another thing that's on, if you're in the Whitsundays region, I'm actually going out on this boat tomorrow. Our tourism operators go out and do some reef health surveys using the Great Barrier Reef Marine Park Rapid Health Impact Surveys and Eye on the Reef and Tourism Weekly survey. So I'm going out with them tomorrow to see what it's all about. But they're running on Wednesdays and Thursdays for the rest of the month. If you are interested, please email deb@redcatadventures.com.au to see how you can jump on. If you forget that email address, just let me know and I can pass that on to you. And the World Science Festival is running in Brisbane, 22nd to the 26th of March, with Curiosity from the 22nd of March to the 2nd of April. We're not actually going to be there, but it's always an amazing event. So jump on to their website to find out how to get tickets and what's going on. And this is an event that's organisation is in progress. We're hoping to have another stand-up paddleboard cleanup event on the 9th of April. As part of a great Easter Trasher Hunt. So follow us on socials. That's the Reef Check Australia Facebook page, or sign up for our newsletter and we can let you know more details as they become available. Sundays with Your Mate. So this is raffles at Your Mate's Brewery in Warana. We do this generally about every 6 weeks. Our next one is the 23rd of April. We'll potentially be there on the 9th as well. That's still being sorted out, but come along and listen to some great music, have some tasty beverages and snacks, and buy some raffle tickets to support Reef Check, because all money raised from the raffle goes to us to help support our surveys and activities. If you want to get involved, you can volunteer. We have 2 ways. As I say, we have our ambassadors and we have our surveyors, and all details on the requirements for both of those is on our website. So if you go to the website, check out the frequently asked questions, and there's a section there on ambassadors and survey divers. And of course, follow us on all of these social things: Facebook, Insta, LinkedIn, and sign up for our newsletter. And of course, we'd like to thank all of the wonderful sponsors who make these events possible: Sunshine Coast Council, Townsville City Council, Port of Brisbane, City of Gold Coast, and the Great Barrier Reef Foundation. And we want to thank the team behind these events, because we need these people to volunteer to make these things happen. And this is our lovely Ilya, who normally hosts our events for us. And as I said, she's in Africa. But we really appreciate the effort she puts in. And tonight's speaker is the lovely Asia, which you can see up there in the corner. I'm not going to tell you this because you would have already read it, and I'll let Asia explain a bit more about what she's been up to and what she's got coming up. So I'm going to stop sharing my screen now. There we are. And Asia can share hers.
Hi, everybody. Okay, can everybody see that okay?
Yep, it's all good.
Perfect. All right, well, thank you all very much for coming along, and thanks, Julie, for the intro. So I'd also like to start by acknowledging the Midjambul people of the Bundjalung Nation. They're the traditional owners of the land on which I reside, and I pay my respects to their elders past, present and emerging, and I'll extend that respect to all First Nations people and acknowledge their continuing connection to land and sea. So, is that? There we go. So the Leaf to Reef team, it's quite a big team of people, very multidisciplinary, with researchers from the University of the Sunshine Coast, University of Queensland, Southern Cross University and Flinders University. And the project is led by Dr. Christine Dudgeon, and with Associate Professor Kathy Townsend as the academic lead. And then there's a whole collection of people involved in the various aspects of the research, including a number of PhD and honours students. So I've been involved with the project to manage the biodiversity data and assist with data collection in the field and conduct outreach and engagement activities. So why Lady Elliot Island? Well, Lady Elliot Island is the southernmost island on the Great Barrier Reef, and was the first climate change ARC included in the Great Barrier Reef Foundation's Reef Island Initiative program back in 2018. So Lady Elliot has been identified as an important land and seascape in the face of climate change for 4 main reasons. So firstly, its conservation value, where it's likely to act as a collector of climate refugees from the central and northern parts of the Great Barrier Reef. Second, for its biodiversity. It's in a unique location, which is close to the continental shelf and those upwellings, and that attracts not only reef, but also oceanic species. Third, it is of economic importance, home to the multi-award-winning Lady Elliot Island Eco Resort. And lastly, due to the threats it faces in terms of invasive plant species, coral bleaching, and crown of thorns starfish. So there are 3 main aims of the Leaf to Reef research. The first was to generate high-resolution maps of the island and the reef flat. The second was to understand the impacts of the extensive vegetation project on the island. And the third is to quantify the biodiversity of vertebrate life on and around the island and determine its connectivity to other locations. So for the purposes of today's talk, I'm just going to be focusing on aim 3, but I'm more than happy to answer any questions that people might have about other aspects of the research as well. So the reason that Lady Elliot Island is of particular interest for climate change, here if we start by taking a look at some of the output from a paper that some of our team were involved in a few years back, we can see global predictions of species range shifts, with the blue areas indicating species loss from a region and the red areas indicating species gain. So this is under 2 different climate change scenarios: business as usual on the bottom and aggressive mitigation strategies on the top panel. So as you might expect, in both of these scenarios, the tropical areas are predicted to lose species, while the subtropics and temperate zones will gain species. So now the same team members that were involved in this paper also ran this analysis in high resolution for the Great Barrier Reef. There we go. And it revealed that the area around Lady Elliot Island, located in the black circle here, is expected to either gain species or maintain its species diversity under future climate scenarios, meaning the waters in this region may represent climate refugia for thermally driven range shifts of mobile species in response to climate change. So the first part of this aim is focused on the biodiversity that's found around the island. So to monitor change into the future, it's vital that we understand what's there already. But despite Lady Elliot being a popular tourist destination and part of very well-researched Great Barrier Reef Marine Park, there wasn't a species list for the island and surrounding waters. So that's what our team set out to create back in 2020 when we started the project. And we're using a variety of approaches, including dedicated research surveys, accessing online databases, and encouraging citizen science submissions so that we can get our best overview of what's there now so that we can monitor change into the future. So just an update of where we're at with this. Bearing in mind that we're just focusing on vertebrate species, as no offense to all the coral people in the audience, we just didn't have the capacity or expertise to tackle the invertebrates in our team. We probably should have hit you guys up for that. But so yeah, this is just a bit of a breakdown of what we've got in our catalogue at the moment. We're close to about 600 species, with the vast majority of these being the teleosts or the bony fish. And we're working on some pretty cool outputs from this work. So one of the outcomes of compiling this biodiversity list for the island is that we're intending to create a field guide for the island and surrounding waters. And this is with the help of some fairly legendary fish nerds, Mark Erdman and Jerry Allen. And by bringing Mark into this aspect of the research, we kind of it was a little bit serendipitous. We actually brought him along to conduct some help with our Manta Ray tagging last year, and it opened up some discoveries that we never really intended from the research. So Mark is a fish taxonomist, and in between tagging Manta Rays, Mark started looking at other fish on the reef, and he pointed out up to 6 species that he believed may be new to science. So 2 of which have been staring us in plain sight. So these 2 common species of damselfish here that have previously been misidentified as other species, and he thought they deserved further investigation as they're likely completely different species. And so on our recent field trip last month, we collected a number of specimens to conduct morphometric and genetic analyses of the 6 species of interest. And yeah, you can follow our social media and everything for the outcome of these investigations. They're just working that data up now, so that's pretty exciting. Okay, so moving on to part 2 of this aim, which is connectivity. So in order to assess the resilience of Lady Elliot as a potential refugia for migrating species, we also need to know how well connected it is to other places in the Great Barrier Reef and beyond. So for this part of the project, we've selected several key species of interest to conduct tagging and genetic studies to determine the movements and connectivity of these species to other reef systems and or to habitats further south. So today I'm just going to go through some examples of these outputs that we've collected so far. I'll just briefly touch on some case studies, and then, as I said, you're more than welcome to ask any more questions. And I know at the beginning, Julie said that you can reserve questions to the end. I also don't mind if you have something burning, you're welcome to interrupt me in the middle. I think we can keep it fairly casual. So the first example that we'll look at comes from the island's red-tailed tropic birds. So Lady Elliot Island is fortunate to be one of only 2 locations on the Great Barrier Reef where these birds nest, and the other being Rain Island in the far north of the Great Barrier Reef. So our population of interest on Lady Elliot is the youngest and smallest known red-tailed tropic bird population. They colonised the island less than 40 years ago, and they consist of only 6 breeding pairs at a time. So this study, which was led by one of our honours students, Caitlin Ivey, compared genetics between Lady Elliot Island and Ashmore Reef to provide insights into the gene flow and connectivity between seabird populations separated by thousands of kilometres. So this study identified a weak signal of population structure between these locations, but also evidence of gene flow, and concluded that these breeding colonies are likely part of a much larger population. The study found really low levels of relatedness and inbreeding within both subpopulations, and this is likely due to high gene flow preventing an inbreeding depression. So these tropic birds are also of interest as they spend most of their life at sea, but during nesting time, they take turns feeding their chicks, and they present this really important source of nutrients for the island. But we actually have no idea where they go during their foraging excursions. So a couple of years back, we deployed some satellite tags on the birds, and we managed to capture this awesome 36-hour track for one of the birds that demonstrated foraging close to the shelf edge in proximity to the mesoscale oceanographic feature of the Capricorn eddy. And this is a real source of productivity to this region. So on to my next case study, and this is the Capricorn silver eyes. So this work was led by one of our honours students, Annika Radu, and she looked at the genetics of these guys, and this is quite a young population as well. So they only colonised Lady Elliot in the 70s, around the time the vegetation started on the island. And we can see here, she has sampled birds from Lady Elliot, which is in the green, Heron Island, which is in the red, and the mainland, which is Moreton Bay, Sunshine Coast in the purple and the blue. And you can see that the birds from Heron and Lady Elliot are distinct populations, but there is one little outlier there between the two, which may suggest that gene flow is beginning between these populations.
Sorry, Asia.
Yeah.
We only got a picture of the bird.
Oh, I'm
there we go.
So sorry.
That's better. Thank you.
I'm so sorry about that. So yeah, thank you for pulling me off on that, Julie. Yeah, so as I said, Lady Elliot in the green, Heron Island in the red, and then the mainland, Sunshine Coast, Moreton Bay over on the purple and the blue on the side there. So they are distinct populations. The island populations are separated from one another, but there is that one in between there that potentially suggests that gene flow may be underway, but they're obviously quite distinct from the mainland ones. So this just suggests minimal connectivity between the reef-based populations and the mainland. Okay, on to the elasmobranchs next, so the sharks and rays. This first example comes with the Epaulette shark. So it is considered one of the top predators on the reef flat. And this species is endemic to the Great Barrier Reef. So this work is the result of an honours project conducted by Jacinta Shackleton, and she was looking at both the genetics of the leopard sharks and the pattern expression on the body pattern of the leopard of the Epaulette sharks. So what she found is that this is just a little bit of the output from the analysis just to demonstrate it. So we can see here the body pattern analysis on the left and the genetic analysis on the right. So she found the body pattern analysis found significant differences between most regions, particularly between the far north, which is in the dark blue, and the south, which is in the red. And then there was a bit of overlap in the middle sections of the central and northern Great Barrier Reef sections. And then the genetic analysis found 4 distinct populations which complemented these findings from the pattern analysis. So it's a really cool little study showing that actually the morphometrics that they display really complements their separation across the reef. Okay, so then the next example are the leopard sharks, and this work is led by Dr. Christine Dudgeon. And for this animal, we actually conducted our field work off North Stradbroke Island, as this is where they aggregate in large numbers during the summertime. So we collected animals on free dive with the very talented help of freediving extraordinaire Kem Cotterill, who some of you may know from MBRS. And the sharks are brought onto the boat and have water pumped over their gills while measurements, genetic samples and bloods are collected. And then the sharks are internally fitted with an acoustic tag with a 10-year battery life for tracking. So just a little bit of background on acoustic tagging. So for those of you that don't know, acoustic transmitters get picked up on acoustic receivers that are located across the reef. So the tagged animal needs to be within range of the receiver, which is about 500 meters to 1 kilometer, depending on any obstructions in the way. And then the receiver needs to be retrieved, the data gets downloaded, and then it is uploaded to the IMOS database to receive information from across the network. So here you can see the extent of the network around Australia, particularly in Queensland, there's hundreds of receivers now in the water. So this is a really important consideration with this data, is that it is presence only, as we don't have any information about where the animal is when they're not within range of one of these receivers. So back to the leopard sharks. What we see from the early movement data shows them moving around the southeast last summer into the spring, with the FAD sites that you can see there on the map corresponding to fish aggregation devices that are located around the Gold Coast. But unfortunately, the data from last year will need to be downloaded from the receivers and then uploaded to IMOS portal for us to be able to update their known movements over the past year. So we haven't actually established the connectivity of these animals back up to Lady Elliot yet. But yeah, watch this space for that one. Okay, so the next example is of detections of Amy, the tiger shark, who was tagged internally back in 2020 off Lady Elliot Island. And the top 4 lines of this graph correspond to detections at sites around Lady Elliot Island, which shows that this tiger shark displays pretty solid residency to the island, with just a few detections showing that she moves down to the Gold Coast on those same FAD receivers that we saw with the leopards. And then the final shark that we tracked at Lady Elliot was the grey reef shark. And here we can see some incredible residency to Lady Elliot. So given its acoustic data, we don't know where the shark is in between detection times, but there are some really decent stretches where it appears the shark doesn't leave the vicinity of the receivers around the island at all. And she hasn't been picked up anywhere else as of yet. Okay, so the last species I will talk about is one of the closest ones to my heart, if any of you know me from Project Manta, so the Manta Rays. So we've been studying their connectivity around Lady Elliot Island well beyond the life of the Leaf to Reef project, but have also gained some pretty interesting recent insights into their movements as well. So one of the reasons for this, these recent insights is that we've been fortunate to deploy some of these really fancy GPS satellite tags that allow for high resolution tracking data. So here you can see the deployment method is by hand spear with the tag attached to the dorsal side of the animal and programmed for an average 100 days attachment. So unlike the sharks, which we can catch and implant internal tags to, manta rays are too big and awkward to do this with. So we rely on external tagging measures, which don't have nearly the same retention time as the internal tags. So here's the output from 5 of these tags, all deployed at Lady Elliot Island over the last couple of years. And I just wanted to have this video play to show an animal with the tag attached to demonstrate that although it may look like the tagging process really disturbs the animal in the last video, they often return to the same behavior within a couple of minutes of the tag attachment. So here you can see the tag just floating above the animal there. So we can see from this tracking data that the majority of the rays spent quite a bit of time in vicinity to the island, but they did move up into the other Capricorn bunker group islands, and some spent quite a bit of time off the northern tip of Gari or Fraser Island in the Long Shoals region, with one spending weeks around the beaches of the northern stretch of the island. And this is quite an exciting finding, as we didn't know that they spent this kind of time in this region. And the tagged ray with the orange track was heavily pregnant. And so we wonder if this points towards potential pupping habitat, which is unknown for this species. Okay, so moving on to some of the acoustic tracking for these rays. There we go. So we tagged 10 manta rays with acoustic transmitters, and in the following months, we saw the tagged animals were picked up throughout the Capricorn bunker group at Heron Island, Northwest Island, and at One Tree, which really complemented the data from the satellite tracking. Okay, so another way to track manta rays is via photo identification using their unique spot patterns. So on their ventral side, they all have unique spot patterns, which allow us to track individuals over time and space via their photographs. So the awesome thing about this technique is that citizen scientists can get involved, which can really extend the spatial extent of records for understanding their range, movements, and connectivity. So using this method, here's just an example of an exciting finding from earlier this year. There we go. So the green lines here represent movements made by some mantas a few years ago between southern aggregation sites, including Lady Elliot, up to the SS Yongala, just near Townsville. And then later last year, we received a sighting of an animal from near Cairns that had previously also been sighted at the SS Yongala. So this is the first record of connectivity between these further north sites and the south, which we'd previously from our data assumed were likely separate populations. And now we think that maybe they could be moving between the two, and we just don't have enough data from up north to confirm that. Okay, and so with that, I hope I've given you a good insight into the different work we've undertaken to address questions around biodiversity and connectivity. And I'd really like to thank all of our funding partners and once again, just acknowledge the large team that's involved in this work, and then open it up to any questions.
That's great. Thank you, Asia. I actually saw 2 manta rays yesterday out at Holburn Island.
Ah, cool.
Yeah, we were so excited on the safety stop.
Any belly shots by any chance?
I don't know. I have to go back and have a look. It was pretty murky. They were feeding in all the like trichodesmium and stuff that was floating on the surface.
Yeah.
So
okay.
I jumped because we saw them on one on the safety stop, got back in the boat, realized there were 2, jumped back in with snorkel gear and got almost run over by one while I was waiting for the camera. And then I was swimming around trying to take photos on the snorkel, and I'm like, they can swim faster than me with no effort.
Yeah, we do find the water up there, because it can be a bit milky or a bit sedimenty. It can be quite difficult to get IDs.
Yeah, it was a challenge, but it was like, oh, how cool was that? So we were really excited. Okay, so has anyone got any questions? Feel free to turn your microphones on and ask them. Okay. It's amazing. I think you've told everyone giving them all too much information.
Sorry, I
Thank
may
you.
have ran through that a bit quickly.
No,
I was going
that's-
to say no questions, but actually that's super interesting. So thank you for sharing, especially with the Epaulette sharks. I found that actually super fascinating that that in terms of the patterns, how it's so congregated in terms of 3 different groups. There are 3 different groups I think I had. It was really
Yeah, potentially 4 with the genetics. It shows with the patterns it was a little bit more mixed up in the middle there with the central GBR, but yeah, it's so interesting. It's really nice to have that complementary data set between the genetics and the patterns.
Yeah, that's super cool. Jules, can you read the comments? There's one coming in.
Yes. Amanda wants to know, are the tropic birds breeding successfully?
Yeah, so every year since we've been monitoring them, and our monitoring began in about 2020, but we have been basically adding to an existing database where they've been monitoring these animals since 1998. Every year there seems to be an average of about 6 breeding pairs, and a number of them are successful at having babies each year. Yeah, so each time we go, they don't have the same kind of seasonality that the other birds do. Most of the nesting birds on Lady Elliot tend to be there during the summer months. But the tropic birds seem to be there kind of year round, with even maybe winter being a little bit busier than the other months. And yeah, every time we're there, there's either birds on nests sitting on eggs, or there's little tiny marshmallow fluffy chicks sitting there, or huge big fledglings that are bigger than the parents getting fattened up and ready to take off. So yeah, they are definitely breeding successfully. It's such a small population, but yeah, as the genetics suggest, we think that it's likely connected to other populations. So has the population there increased? Well, not since we've been monitoring it. So it seems to be an average of 6 nesting pairs since they started nesting there, which was around 30 years ago. So I think I may have shown in one of the slides, I'll just go back if I can. It's really cool. This bird, which we coined Whoopi Old Bird, is actually a male bird, we now know, because its partner laid the egg last season. But he was tagged as a fluffy little chick back in 1998 or 1997. So we now know he's like a 25-year-old bird. So it doesn't seem that, it doesn't seem that the population of nesting on that island is increasing. But in terms of the broader population, we're not sure. We are working with other researchers to understand that a bit better.
Yeah, it's interesting that that the number of breeding pairs has stayed consistent.
Yeah.
And I suppose that's another research project, is to find out why it's staying at sort of 6 breeding pairs.
Yeah, it's interesting because, as I said, on the Great Barrier Reef, the only other place where they nest is Rain Island. So it's a long way away. It's not like they're nesting all across the Capricorn bunker group or something, in small numbers. They're nesting in small numbers in this one place, and then it's thousands of kilometers until the next one. There's some in Lord Howe Island, some Norfolk Island, Rain Island, up across at Ashmore Reef over, off the Kimberley. So it's actually, it's quite a distance between these populations. Some of those other locations have far more birds than Lady Elliot, but Lady Elliot's is, quite small.
Okay. Karen would like to know, are you doing any study around the turtles at Lady Elliot?
Yeah, so one of the PhD students that's been involved in the project, I didn't actually include it here because the focus is not so much on connectivity, but she's been studying toxins on the turtles. So she's been coming over to the island, and we've been collecting, I believe she has collected about 30 juvenile green turtles and 10 juvenile hawksbills. So she's particularly interested in the juvenile stages because they're the ones that are there feeding predominantly. So the adults use the location for nesting, but the juveniles are there feeding. And so she's interested in the toxins in their system to see how healthy they are, to see if they're ingesting plastics and pollutants and that kind of thing. So yeah, that's the main focus of her work there.
How is she doing that? Is she making them regurgitate? Oh, upstairs she's not killing them, but
no. So so no stomach contents analysis. She actually takes blood samples and she measures the toxins in their blood. Yeah.
Okay. Yep.
Yeah. So, and she also does the tagging, like with the Queensland Parks and Wildlife Service, is the same titanium tags that they use. So she has been flipper tagging each one that she collects, and she takes a whole pile of measurements to do with their size and their condition and everything to go along with the blood work.
Okay. I know Karen's got a vested interest in turtles. She's one of our Sunshine Coast turtle care leaders.
Yeah, no, I know Karen as well. We met we met a number of years ago, and then again, was it last year on Lady Elliot, I think, Karen?
Yeah, it was last year, I think. Yeah.
Yeah, yep.
I love the turtles around there. So many in the lagoon
friendly.
there.
So
Yeah.
friendly.
I always go up with a data sheet and get all the tag numbers and send them through to Cole.
Oh, nice. That's awesome. Yeah, no, well, I can tell you, I had not had experience with researching turtles until being involved with Caitlin's work for her PhD. And so I wasn't so much involved in taking the measurements or anything, but I was one of the in-water turtle wranglers. And Lady Elliot is a very easy place to be a turtle wrangler. You literally just sit in the water and they go, "We need another turtle." And you go, rather easy.
That sounds great.
Beats jumping off a boat with a helmet on, doesn't it?
Definitely does. Thanks, Asia. That's fascinating.
Yeah, no worries.
All right. So
yeah, I wanted to cover try and cover the breadth of the connectivity work. But I realized for many people, I probably just touched on it, and some people probably have a key species of interest that I went far too quickly past. But you can always hit me up with more questions.
You didn't even count the nudibranchs. I could have done that for you.
I know. I thought that I'm giving this talk to a bunch of reef checkers that are going to be so offended that we didn't even try to categorize the corals.
Oh, corals can be hard. That's all right. I think we've got survey sites on Lady Elliot, so we got 2 last year, so hopefully again soon.
We do. We've got 5 sites there, so with any luck, we will manage to get back over there sometime. It's a very busy place though, so.
Oh, I know. We find it hard to get accommodation, and we're partnered with the resort.
Yeah.
What hope have we got? Oh, well, that was amazing. Oh, so that's the little silver eyes. So the student that did that study, so she also did genetic sampling like in Moreton Bay and Sunshine Coast and Heron Island.
Yeah, so I don't believe she did the sampling at Heron. I think it was a part of a collaboration. So that's one of the things with this project. I will confess quite honestly, Leaf to Reef runs on a very tight budget. And so all of these things that I think can look like we've done a huge amount. It's why I declared at the beginning and at the end, this is part of a huge collaboration. So when we wanted to investigate connectivity, immediately the question was who's doing what where, and how can we work with our networks so that we're not reinventing the wheel and we're not, exhausting research budgets. So we'll work with who's doing things, and we'll collaborate with them help them address their questions, help us address our questions, and just maximize the work that we can achieve.
Yeah, beautiful. I was thinking that's a lot of work for an honours project.
Yeah, exactly. Yeah.
Yeah. Amazing. So does anybody else have any other questions? No? Well, in that case, we might leave it. Obviously, if you've got any burning questions that you think of later and you'd like me to pass on to Asia, just send me an email at seqsurveys@reefcheckaustralia.org and I can pass them on for you. And I would like to thank you all for coming, and I'd like to thank Asia for doing this presentation for us in a BP service station somewhere on the highway. It's actually a pretty quiet service station.
Wasn't too bad.
Right, so I'm going to...
Nailing it, mate. Good work.